Factors affecting enzyme activity. So just to see where this comes in the topic of biological molecules, done monomers and polymers, done carbohydrates, done lipids. We've looked at the structure of proteins, and we're now looking at how many proteins are enzymes. We've also done the induced fit model and the specificity of enzymes, so we're now looking at the effects of the following factors on the rate of enzyme-controlled reactions: enzyme concentration, substrate concentration, concentration of competitive and non-competitive inhibitors, pH and temperature. So, ways of measuring enzyme activity. So the rate of a reaction can be measured in two ways. It can either be how fast the product is made. So there are new products produced that are different from the reactants. By measuring the amount of end product present at different times, the reaction rate can be calculated. So an example of this would be when you add catalase to hydrogen peroxide, you will get oxygen bubbles produced, and you can see how much of that oxygen is produced in a certain length of time. You can also do how fast the substrate is broken down. So to make the products, the substrate molecules have to be used up. By measuring the amount of substrate molecules left at different times, the reaction rate can be calculated. So we'll look at some experiments and some graphs. So first of all, we'll look at the effect of temperature on the rate of enzyme-controlled reaction. So first of all, we want to know the difference between describe and explain. So if we were to describe this graph, you literally just look at the patterns. You don't explain why, you just say what is happening. So from 0 to 40 degrees C, as the temperature increases, the rate of reaction increases. And the optimum is just under 40, about 39 degrees C. Then you could say from 45 to 60 degrees C, as the temperature increases, the rate of reaction decreases. So that's just literally describing it. Now, to explain it, we need to say why that happens. So, the rate from 0 to 40, the rate of reaction increases because as temperature increases, because more heat means more kinetic energy, so the molecules move faster. This means the substrates are more likely to collide with the active sites. The energy of the collisions increases, meaning each collision is more likely to result in a reaction. Okay, and then if we look at this part of the graph, if the temperature gets too high, the reaction stops. The rise in temperature makes the enzymes vibrate more. Eventually, the vibration breaks some of the bonds that hold the enzymes in shape, such as hydrogen bonds. The active site changes shape, and the enzyme and substrate no longer fits. At this point, the enzyme is said to be denatured. You can see that at low temperatures here, the substrate fits into the active site, but there's probably not very much kinetic energy. At high temperature, the enzyme vibrates more. This breaks the hydrogen bonds that hold its shape. And then at very high temperatures, the active site changes shape, and the substrate can no longer fit, so the enzyme is denatured. So just to point that different enzymes have different optimum pH, optimum temperatures. So for most human enzymes, it's around 37 or 38 degrees C. But some enzymes, like those in biological washing powders, the optimum is 60 degrees C. So now if we look at the effect of pH. All enzymes have an optimum pH value. Most human enzymes work best at pH 7, which is neutral, but not all of them. So for example, pepsin works best at pH 2. It works best in acidic conditions, which are found in the stomach. Above and below the optimum pH, the H plus and OH minus ions disrupt the ionic bonds and the hydrogen bonds that hold the enzyme tertiary structure in place. So the enzyme denatures and the active site changes shape. Effect of substrate concentration. So you can describe this graph first of all. So you can see that as the substrate concentration increases, the enzyme activity increases up to a certain point where further addition of more substrate has no additional effect on the enzyme activity. Okay, so now looking at explaining why. So the higher the substrate concentration, the faster the reaction from here to here, because more substrate means collisions between substrate and enzyme are more likely and more active sites will be occupied. But then, up until a saturation point, after which there are excess substrate molecules and all the active sites are full, so adding more makes no difference. So low substrate concentrations, not all the active sites are occupied. You've got a saturation point, which is where all the active sites are occupied. But beyond that point, the saturation point, all active sites are already occupied. So adding more substrate won't make any difference. Okay, so now we'll look at the effect of enzyme concentration. So to describe it, first of all, you'd say as enzyme concentration increases, the rate of reaction increases up until a certain point where adding more enzyme has no additional effect on the rate of reaction. And now to explain it, the more enzymes there are, the more likely a substrate molecule will collide with one and form an enzyme-substrate complex. Increasing the concentration of enzyme increases the rate of reaction. And then here, if the amount of substrate is limited, then there's a point when there's excess enzyme molecules and not enough substrate. So adding more enzyme has no effect. So basically the substrate is a limiting factor. So it doesn't matter how much enzyme you have, there's not enough substrate. It's really important when you're looking at an exam question that you read the axes, because not all graphs are rate graphs. Some of them might be volume of product. So, for instance, this one, describe and explain the three curves shown below. So, the first thing you would do, you would compare the rates of reaction at the start of the graph. So the rate of reaction was fastest at the start at 65 degrees C, because the curve is steeper. And then you could say the reason why, you're explaining it, this is because the molecules have more kinetic energy, so they are moving faster, so the substrate is more likely to collide with an enzyme's active site. More enzyme-substrate complexes are formed, and more energy means collisions are more likely to result in a reaction. Then you would compare the different temperatures at other points. So at 37, the graph has plateaued after 40 seconds. And this is because all the substrate has been used up. At 65, the graph has plateaued a lot earlier. It's plateaued at about 10 seconds. And that is because the high temperature has caused the enzyme to denature, so the reaction has stopped. And therefore not as much product was made. That's why it's not gone as high, because not all the substrate was converted to the product before the enzyme was denatured. At 25, the rate of reaction is remaining constant and the volume of product is continuing to increase, but not all of the substrate has been used up because it isn't going fast enough for the reaction. Okay, now looking at a worked exam question from AQA. So a technician investigated the effect of temperature on the rate of an enzyme-controlled reaction. At each temperature, he started the reaction using the same volume of substrate and the same volume of enzyme solution. Give one other factor the technician would have controlled. So he's investigating temperature on the rate of reaction. So it means he's going to have to keep everything else the same. Now he's already said that he's going to use the same volume of substrate and the same volume of enzyme. So you've got to keep all other factors the same. So you definitely keep the pH the same. You keep the concentration of the substrates the same, the concentration of the enzyme solution the same. Calculate the rate of reaction at 25 degrees C. Okay, so 25, you do y over x, so it would be 10 divided by 240. So you would get 2.5 grams per decimeters cubed per minute, or 0.04 gram per decimeter cubed per second. Describe and explain the differences between the two curves. So you get a mark for your answer, a mark for your units there. The units is always the y over the x. Describe and explain the differences between the two curves. So the initial rate of reaction for 37 is higher or faster because there is more kinetic energy. So there will be more enzyme substrate complexes formed, and the graph reaches a plateau at 37 because all the substrate has been used up.